Hi, I'm Sam Hall from Pharmacodynamics. In this video we're going to talk about the kinetics of drug-target interaction. Here we're talking about time, the time it takes for the drug to recognize the target, which is the association process, and how long the drug remains bound to the target, which is governed by the dissociation process. This seminar will focus on the impact of binding kinetics on drug discovery. We'll do this from bedside to bench, from the impact on drug effect in man to the impact on the in vitro assays used to discover new therapeutics. Let's first go through a brief introduction on the basics of binding kinetics. Here's a cartoon of a simple drug-target interaction. Drug, in red here, associates with the target in blue. This is governed by the association rate constant, or K-on. The drug and target then stay together for a period of time, then the complex dissociates, governed by the dissociation rate constant, or K-off. Now this dissociation rate is of considerable interest in drug discovery. It defines the average length of time the drug remains bound to the target. This length of time is usually expressed as the residence time, which is equal to 1 divided by K-off. A long residence time could help drug efficacy, which is good, but it can also introduce artifacts into the in vitro assays we use to optimize new therapeutics, which is bad. We'll be going through this today. One final bit of theory. The binding kinetics are related to the binding affinity. Kd, the affinity, is equal to K-off divided by K-on. This will be important later. The best way to understand binding kinetics is to see it in action. With the advent of single molecule spectroscopy, this is now possible. Experiments have been done with fluorescent ligands, which emit light when they are bound to the receptor, as shown here. A really nice example of this technology is provided in this paper. You can get this link by downloading the PowerPoint slide deck used to make this video, which you can get from the link provided in the comments section. Now let's see what this looks like. Here we have a population of receptors. Now let's turn the lights off. Now when a ligand binds to the target, we'll get an orange circle appear like this, indicating the ligand has bound to the target and is fluorescing. When the ligand dissociates from the target, it will no longer be visible and so will disappear like this. First, we'll show the association process. On the right here is a graph showing the number of receptors bound by the ligand over time. Now we're going to run the simulation. The graph here shows the time course of binding. This is the classic exponential association curve. Binding first increases very rapidly, then slows down, then it finally reaches a plateau where binding is at equilibrium. From the simulation, you might have noticed that equilibrium isn't static. Instead, drug is continuously associating and dissociating. Now let's look at dissociation. In this experiment, we preload the target with the ligand, so we start with a population of ligand-bound targets, indicated by the orange circles here. Now we can simulate the dissociation process. The graph here, as shown in the graph, and as you will have noticed in the animation, dissociation of the population first starts very rapidly, then slows down, then gets much slower as the number of ligand-target complexes approaches zero. This is defined by the classic exponential decay curve. Now from this curve we can quantify the residence time. We fit the data to an equation, and this gives us the fitted value of K-off, from which we calculate the residence time. Here the residence time is 3 minutes. Now the residence time can vary. Different ligands can have quite different residence times. Here's a dissociation curve for a shorter residence time of 1 minute in green here. And here's what the data look like for a compound with a longer residence time of 15 minutes. Binding kinetics has become of great interest to drug discovery for reasons we'll be seeing today. Here are some of the primary reviews that provide a good introduction. Throughout this presentation, you'll see links to key papers if you want to take a deeper dive on specific topics. The first thing to note is that residence time can vary enormously. Here is a sample of the residence time of numerous drugs in clinical use. Note this scale at the bottom here goes from 1 second to 1 week. On the short end of the scale, we have a couple of ion channel modulators here with residence times of around a second. Then we can see that the majority of drugs have residence times from around a minute to around an hour. Then some compounds bind with a long residence time of many hours. Finally, some compounds form extraordinarily long-lived complexes with their targets, with residence times of several days. Some of the long residence time compounds are renowned for having unusually strong efficacy or for having a prolonged duration of action. That completes the introduction. Here's the outline for the rest of the presentation. Basically, we're going to talk about the why, the how, and the when of binding kinetics in drug discovery. Why should we think about kinetics? I'll go through 2 reasons. First is that kinetics can impact drug effect in man and in animal models. Here we're talking pharmacodynamics, and I'll show you how binding kinetics intersects with pharmacokinetics to control drug efficacy. The second reason is that the in vitro assays we use to discover new therapeutics can be affected by binding kinetics. These are the assays we use to define SAR during lead optimization, for example. It turns out that a long residence time can profoundly distort the measurement of drug potency, which can cause a lot of problems, as we're going to see. Then we'll go through the methods we use to measure the kinetics of drug binding. There are 2 approaches. First, we'll go through the assays that detect whether our compounds have a long residence time, since this can be a benefit or a problem. Second, we'll go through assays to actually quantify the rates of binding. We'll bring all this together using a real-world case study I was involved in, where we applied binding kinetics to the discovery of antagonists for G-protein coupled receptors. We'll finish with some thinking about when to apply kinetics in the drug discovery process, given that this takes significant resources. Let's start by looking at the pharmacodynamics. Here we're going to talk about the intersection between pharmacokinetics and binding kinetics in defining drug effect. This is where the action is for the value of kinetics to drug discovery. We'll start with the simplest case. One of the main arguments for the kinetics approach is that a long residence time can compensate for lack of compound stability, in other words, rapid compound elimination. A standout case is provided by the drug opicapone. This is a new drug for Parkinson's disease. Opicapone stabilizes the levels of L-DOPA by inhibiting the enzyme that metabolizes it, catechol O-methyltransferase. Once-a-day dosing is the goal here. In this experiment, healthy volunteers were dosed with a single dose of opicapone. In red here is the drug concentration in the plasma over 24 hours. You can see the drug doesn't last long. The half-life is 1.2 hours, and by 24 hours the drug has been completely cleared. Now let's bring in the drug efficacy. Here we're looking at target engagement by measuring inhibition of activity of the target, the COMT enzyme. In blue here is the target engagement over 24 hours after the dose. We can see clearly that engagement is sustained throughout the day here, and that the drug is effective long after the free drug has been cleared from the circulation. This experiment was extended out to 6 days after the dose was given, as shown here. Remarkably, the target was engaged by the drug 6 days after dosing, long after the free drug had been cleared. This is explained by opicapone having a very long residence time for COMT. In vitro, the residence time was measured to be 4.2 days. This agrees pretty well with the PD half-life of 5.4 days. So this is a really nice example of how residence time can impact pharmacodynamics of a drug in clinical use. Now this is an example for a single dose of drug. Now let's look at what happens when we give multiple doses of drug. To do this, we're going to use a simulation of a theoretical drug. This simulation will show how a long residence time can enable a lower dose to be used in a repeat dosing paradigm. Here we have a drug dosed once a day for 7 days. In red here is the drug concentration in the body. In this example, the compound pK has an elimination half-time of 4 hours, so this compound is moderately stable. We can see the classic peak and trough profile here, and this is repeated over 7 days. Now let's bring in the target engagement. Let's suppose we want the drug to occupy at least 60% of the targets over the 24-hour dosing interval. That's indicated by the dashed blue line here. First, let's consider a drug with a short residence time of 5 minutes. We assume the drug affinity is 30 nanomolar. Now we need enough drug to get at least 60% target engagement across the 24 hours. This requires a dose of 10 milligrams and a peak concentration of 1400 nanomolar. Now there's something to notice with this profile. In order to get sufficient occupancy at the trough, we need to force up the drug concentration at the peak. This is because the drug concentration is declining over time. In order to get sufficient drug on board at the trough, we need a lot more drug at the peak. Here the peak concentration is 1400 nanomolar, which is in large excess over the affinity of the drug, which is 30 nanomolar. The upshot is we are giving much more drug than we need at the peak in order to get sufficient target occupancy at the trough. This might be a liability. Oversaturating the target could be problematic if there is mechanism-based toxicity, and overdoing the dose could be a problem if there is a therapeutic window issue and off-target activity concerns. Now let's look at a compound with a long residence time of 36 hours. We're going to use the same y-axes as we used before. We assume the affinity of the compound is the same as that for the short residence time drug at 30 nanomolar. We also assume compound absorption and elimination is the same as that for the short residence time drug. So how much drug do we need to stay above our 60% target occupancy threshold for our long residence time drug? We need much less, as it turns out. Here are the simulated data for the dose necessary to achieve 60% occupancy. We need a dose of 1.4 milligrams, which is 7-fold less than the 10 milligrams we need for the short residence time compound. This lower dose requirement is also evident when we look at the compound concentrations at the peak. For the long residence time compound, we need 190 nanomolar, which is 7 times less than the 1400 nanomolar we need for the peak concentration for the short residence time drug. This is good news if we have a selectivity issue. Lowering the drug concentration could lower occupancy of unwanted targets. This could be beneficial for a kinase inhibitor, for example. The other thing to notice is that we are occupying fewer targets at the peak. Here we're at 77% occupancy at the peak versus 98% for the short residence time compound. This could be valuable to avoid mechanism-based toxicity associated with the target. This concept is known as kinetic selectivity, and it's been highlighted in numerous papers, as shown here. Now I've shown you a snapshot of when a long residence time can be beneficial. The extent to which this is true is dependent on the relationship between the compound pharmacokinetics and the binding kinetics. For example, when the compound resides in the body a lot longer than it resides on the target, the target residence time doesn't have much of an effect. Now, there are a lot of potential scenarios, and these papers here are an excellent resource for those of you who want a deeper dive. We also need to think about short-acting drugs. Sometimes we want a compound to have an effect for a certain duration of time before the effect wears off. Here we're talking about sleep drugs, emergency medicines and anesthetics, for example. Here a long residence time can be a problem, as you can imagine from seeing the last few slides. Let's consider a sleep drug. We want rapid onset of action to improve sleep latency, then we want efficacy to persist for a certain period of time, and then we want the drug to wear off so as to minimize impairment of next day performance. The classical way to do this is to tune the elimination rate of the drug by controlling its PK. Here's a PK/PD profile similar to that of the GABA-B modulator Ambien. In red here we have the drug concentration with an elimination half-time of 2 hours. In blue here we have the target engagement for a compound with a short residence time of 5 minutes. Target engagement of 30% is required for efficacy for this class of drugs. We hit this threshold in a few minutes with this compound, remain above it for about 5 hours, after which the target engagement wears off. So this timing is just right for a sleep drug. Now let's consider a slow residence time drug, which is this dashed blue line here. Clearly things are way off. This compound has the same affinity as the first one, but has a much longer residence time at 6 hours. The first thing to notice is that onset is very much delayed. We're not reaching threshold until 3 hours. This is actually anticipated. Long residence time drugs take longer to equilibrate with the target, and this has a sound basis in theory, and we will be coming back to it later. This delay means the drug won't help with sleep onset. The second thing is that the duration goes out to 7 hours, which will produce next day impairment effects. Now this simulation was done for human efficacy, but the concepts here also apply to acute animal models. Imagine running an in vivo assay where efficacy is measured at 1 hour post-dose. A slow off-rate compound will be much less active than a fast off-rate compound, even if they have the same affinity. This means that the slow off-rate compound could be disregarded, even though on chronic dosing it could be the best molecule in terms of efficacy. Based on this thinking, it might be a good idea to avoid early time points in vivo, or to take a second time point later in the time course. Now at this point it's worth doing a thought experiment. Imagine if we didn't know the residence time on the human target. We take our sleep drug into a clinical trial and it has this quite unexpected PK/PD profile, because the compound has a really long residence time. This is an expensive way of finding out the residence time issue. A much cheaper way is to check the residence time much earlier, say at the advanced lead stage, and we'll be talking about how to do that soon. Now there are some additional factors to consider. What we've seen so far does not consider the endogenous ligand for the target. This could be the endogenous agonist for a receptor or a substrate for an enzyme. The endogenous ligand concentration can change over time. Examples include synaptic release of a neurotransmitter, shown here, which takes milliseconds, release of a hormone like adrenaline, which happens over minutes, or diurnal rhythms, such as corticosterone, which can take hours. When the endogenous ligand fluctuates, interesting things can happen, as we're about to see. Now this is very much under-investigated, and all we have right now are simulations. I'll walk you through a simulation here. In this case, things are a bit different from the previous slides. Here we assume we have a constant level of drug. This constant level of drug produces a constant level of occupancy in the absence of the endogenous ligand. This target occupancy by drug is indicated by the blue line here. Now let's bring in the endogenous ligand. Here in red is the concentration of the endogenous ligand. The concentration changes over time, We have a steep rise, a peak, and then a decline back to zero. Now suppose this drug in blue here has a really short residence time of 10 seconds. What happens under this condition is the drug gets displaced from the target by the endogenous ligand. Because the drug comes off the target so quickly, the target gets freed up and is swamped by the endogenous ligand. This means the drug is no longer very effective at occupying the target. The trough occupancy by drug is just 16%. Now let's consider a drug with the same affinity but a long residence time of 60 minutes, as shown here. Here is the occupancy in the absence of the endogenous ligand, which is the same as before. Here is the endogenous ligand pulse, as before. And now here is the occupancy by the drug in the presence of the endogenous ligand. What we are seeing is that the occupancy remains sustained in the face of the endogenous ligand pulse. The trough occupancy is 83%. This happens because the drug doesn't dissociate very much from the target, so most of the targets remain occupied by the drug over the pulse of the endogenous ligand. Now this isn't a black and white phenomenon. For the short residence time ligand, we can get effective block by increasing the dose, as shown here. We have a short residence time of 10 seconds, but have increased the dose 25-fold. This gives us the same level of occupancy at the trough as the slowly dissociating ligand at 83%. Here we have sustained block simply by swamping the system with drug, blocking the binding of endogenous ligand by simple mass action competition. But it takes 25 times as much drug to get the same effect as a long residence time. This means a long residence time could potentially enable a lower dose, potentially improving the therapeutic window. Here we have taken a few snapshots of binding kinetics and pharmacodynamics. There is a large array of potential PK/PD scenarios. To help investigators explore their own conditions of interest, we have made some simulators. These are super simple to use. They are just Excel spreadsheets. You enter the parameter values and the graph gets updated. The simulators are free and are available at the link given here and in the comments section. All the examples you have seen here today were made with these simulators. Now, if we are going to apply binding kinetics to clinical drug development, we must consider the logistical realities involved in advancing a drug to clinical trials. The rubber hits the road when we come to predict human dosing. Now, we've gotten pretty good at this. Not many compounds these days fail for PK, for example. This relies on PK/PD modeling, and the simple fact is that the large majority of this work does not consider binding kinetics. As a rule, PK/PD models usually assume a steady state between the free drug and the bound drug, and that drug effect is driven by compound concentration. We need to think about what the consequences will be of including residence time here. It adds a variable to the equation, and this adds uncertainty. Specifically, we don't know how residence time in a test tube compares with residence time in a living organism. For this reason, an early and robust read on target engagement in man would be highly desirable. There are some examples where clinical PK/PD modeling has incorporated binding kinetics. Some really nice work on this has been done on opiates such as buprenorphine. Now we're going to turn our attention to the in vitro phase of drug discovery. Here we're talking about the target-based assays we use to quantify compound potency to establish SAR and optimize new molecules. We're going to see some scary stuff. It turns out that a long residence time can, in certain circumstances, really mess up with these assays. Specifically, a long residence time can result in dramatic underestimation of the affinity of our compounds if these assays are not incubated for long enough. Now, this is based on one of those little details that pharmacologists think about. This detail is that drug affinity is an equilibrium parameter. Affinity is the concentration of drug required to occupy 50% of the targets at equilibrium. Now it takes time for equilibrium to be reached. When you look into the mechanics, something quite surprising emerges. The time it takes to reach equilibrium is dependent on the residence time. Compounds with a long residence time take a long time to reach equilibrium. For such compounds, if we don't incubate the assay for long enough, we will underestimate their affinity for the target. Let's see this in action. In this experiment, we are measuring the affinity of a compound binding to a target using a standard binding saturation assay. This is a compound with a long residence time of 6 hours. The affinity of this compound is 1 nanomolar. The black line on the graph here shows the true affinity of the compound. Now let's assume an incubation time of only 10 minutes. Here's what the data looks like. Something is clearly wrong here. The affinity is 1 nanomolar, but the midpoint here is 20 nanomolar. So this assay isn't reporting the affinity correctly. With a short incubation time, we're dramatically underestimating the affinity. Now let's increase the incubation time to 1 hour, something we might use in a binding assay. This assay is still not reporting the affinity correctly. The measured affinity value here is 4 nanomolar. It turns out we have to increase the incubation time out to 12 hours for this assay to report the affinity correctly, shown by the data in red here. Simulations and experiments have shown the incubation time needs to be at least 2 times as long as the residence time for assays to correctly quantify the affinity. One message from this slide is that we should avoid really short incubation times for our SAR assays. Under this condition, we can miss the winners. Here we can't tell the difference between a 1 nanomolar compound and a 20 nanomolar compound at 10 minutes. This artifact is based on the mechanics of ligand association with the target. Here is a graph showing the time course of target occupancy by the compound from the previous slide. This is shown for various concentrations of the compound, from 100 nanomolar down to 0.032 nanomolar. When equilibrium is reached, the curve approaches a plateau, which is on the right-hand side here. The thing to notice here is the time it takes equilibrium to be reached is dependent on the concentration of compound. Equilibrium for the highest concentration, 100 nanomolar in blue here, is reached within a few minutes. By contrast, for the lower concentrations, for example 0.32 nanomolar here, it takes a lot longer. It takes 12 hours for this curve to reach the equilibrium plateau. It is this that distorts the binding curve we saw on the previous slide. We can see this more clearly if we put the time course and the dose-response curves side by side. If we take the 10-minute time point right at the start here, we can see that while the highest concentration has reached its equilibrium plateau, the lower concentrations are nowhere near their equilibrium plateau values. This means occupancy for these lower concentrations is much less than the equilibrium value. Now we go to the occupancy graph on the right and we plot these 10-minute values, shown in blue here. We can see again occupancy is underestimated at the lower concentrations. It is this that results in the curve being shifted over so far, with the result that the affinity is underestimated. Now let's look at the 1 hour time point, shown here. We see a similar but less pronounced effect, and on the occupancy graph there is a smaller shift. Finally, at 12 hours, we have closely approached equilibrium for all concentrations. The association curves have all reached their equilibrium plateau values, and the occupancy curve on the right reports the true affinity of the compound. Now this is just a single compound. What happens when we have a range of compounds with a range of residence times? It turns out this underestimation of affinity is dependent on the residence time of the compound. Here we're going to simulate this. Here we have a range of compounds, each represented by a separate circle. These compounds have a range of affinity. The affinity is on the y-axis here. Now in this example, we assume the affinity is determined by the residence time. The longer the residence time, the higher the affinity of the ligand. So we have a compound at the top here with a residence time of 48 hours and an affinity of 23 picomolar. The compound at the bottom has a residence time of 11 minutes and an affinity of 12 nanomolar. Now the data we see here assume the assay is at equilibrium. Let's now assume the incubation time is 90 minutes. Now at 90 minutes we are nowhere near equilibrium for these compounds at the top, but we are at equilibrium for the ones at the bottom. Remember, we need an incubation time that is double the residence time for the assay to approach equilibrium. When we look at the data here, clearly things are way off. The affinity of the long residence time compounds is dramatically underestimated. The 48-hour residence time compound here has a measured affinity of 690 picomolar compared with a real affinity of 23 picomolar. This underestimates affinity by 30-fold. For the shorter residence time compound of 11 minutes, the 90-minute incubation reliably quantifies the affinity at 12 nanomolar. When we look at the data set as a whole, we can see at 90 minutes we hit an assay floor. The compounds all bunch up around the 1 nanomolar mark. This means compounds could be a lot more potent, but we would not be able to detect them using the 90-minute assay. This region here represents a lot of SAR we wouldn't be able to detect using this assay. This can do a lot of damage to drug discovery campaigns. Often the best compounds go unnoticed because we're not incubating long enough to detect their high affinity. This results in missed SAR opportunities and failure to test the best compounds in vivo. It can even impact human dosing because the compound could be a lot more potent than we think it is. This artifact happens because the residence time determines the time it takes for equilibrium to be reached. This rather surprising fact is shown on the association graphs here. On the left, we have a long residence time compound from the previous slide of 6 hours. On the right, we have a shorter residence time compound of 23 minutes. For the long residence time, it takes a long time to reach equilibrium. Here we have the 6-hour time point, and we're still not at the plateau for the lower concentrations. Contrast that with the short residence time compound on the right. Here the equilibrium plateau is reached within 1 hour for the low concentrations of ligand. It is this surprising dependence of equilibration time on residence time that underlies the artifacts that distort the measurement of affinity if the incubation time is not long enough. Now, having gone through the benefits and liabilities, we come to the methods that can be used to evaluate binding kinetics. There is an ocean of information on this, and here are some really useful reviews. Here I'll provide just a couple of snapshots. There are a couple of ways of looking at binding kinetics in drug discovery. First is an approach that asks, do my compounds have long residence times? This qualitative approach does not take a lot of resources. Given the potential benefits and liabilities of the long residence time, it's good to have ways of addressing this without going to too much trouble. The second approach is to measure the rates of binding using dedicated kinetics assays. These are usually feasible but require more resources. We'll start with the first approach, detecting whether compounds have a long residence time. One simple way to do this is the so-called Ki shift assay. Here's an example for a receptor, the gonadotropin releasing hormone receptor. What we have here is a simple competition ligand binding assay. Compound is competed against a tracer in a dose-response format. Then this is run at 2 different times, a short incubation of 30 minutes here and a much longer incubation at 10 hours. If the compound has a long residence time, as shown here, the dose response will shift to the left for the longer incubation. In other words, the compound will appear to be more potent. If the compound has a short residence time, as shown here, the curves for the time points will overlie one another. The data can be analysed by calculating the Ki shift, dividing the Ki of the short incubation by the Ki for the long one. Here is a plot of the Ki shift of 78 compounds in the assay. 7 of these had a ratio greater than 6 and were then shown to have a long residence time, in this case 90 minutes or longer. Another quick check is the insurmountability test. This is popular for cell signaling assays and is used for antagonist compounds. Here we have an example of cyclic AMP generation by a GPCR, the receptor for corticotropin releasing factor. We do a Schill type experiment where we measure the dose response to the agonist in the presence of increasing concentrations of the antagonist compound. There is a nice diagnostic pattern that can emerge. Short residence time compounds, like the one on the left, produce a shift of the dose response curve, but they do not reduce the maximal response. These curves all plateau at the same level. By contrast, a long residence time compound behaves differently. The antagonist reduces the maximal response to the agonist. This is called insurmountability, and it can be diagnostic of a long residence time. This isn't always the case though. Allosteric modulators can also produce this insurmountable behavior. If we figure out there is a residence time issue on the target, the next stage is to measure the binding rates K on and K off. The best way to do this is with a binding assay. In a perfect world, we would have a direct target ligand binding assay. The most widely applied direct assay is the surface plasmon resonance assay, as shown on this slide. This usually requires a purified and well-behaved target. Here, all we have in the assay is the compound and the target, and the binding signal is recorded as a change in the SPR intensity. We measure this over time. The first phase here is association, then we remove the compound and detect dissociation. Then we fit the data to standard equations to estimate K on and K off. Usually we don't have the luxury of a direct binding assay, and instead we use an indirect competition type binding assay. Here we're inhibiting binding of a tracer compound by our unlabeled compounds of interest. Fluorescent tracers are ideal for this because the assay can be run in real-time format. We put a single plate in the reader and read it repeatedly over time to determine the time course. This graph is a fluorescent tracer binding to BTK kinase, and it shows the large number of time points we can collect with this modality. Tracer binding is measured by itself and in the presence of a range of concentrations of inhibitor test compound. What we do now is fit these data to an equation which returns fitted values of K off and K on of our unlabeled test compound. This is the so-called Matulsky and Mayen method. At the bottom left here are a couple of papers that go into the details. What if we don't have a binding assay? Fortunately, functional assays can be used to measure kinetics of inhibitor compounds. All we need is an assay of the target's function, like an enzyme activity assay or a signaling assay for a receptor. This involves the washout method. Compound and target are incubated together to form the complex. Then the unbound compound is removed by a washout or jump dilution step. After this, the compound can start dissociating from the target. We then add the reagents to measure target activity like enzyme substrate or receptor agonist. When the compound dissociates from the target, activity of the protein commences that we can then measure by product formation or signal generation. Here is an example for an enzyme using the jump dilution method. Here we're measuring ADP release by the protein kinase ABL1. We see the straight line time course for the control without inhibitor. In the other traces, we see the inhibitor slows down the enzyme activity, and this is because the inhibitor first has to dissociate before the enzyme activity starts. An equation is available to analyze these curves to determine the dissociation rates shown in the table here. Here's an example for a cell signaling assay. Here the H1 histamine receptor is incubated overnight with antagonist. The antagonist is then washed away. The cells are then incubated for various times before the addition of histamine. The response is measured a short time after histamine is added. In this case, we're measuring calcium signaling. On the right is the graph. Here, the y-axis is the time interval between washout of antagonist and the measurement of the histamine response. Mepyramine dissociates rapidly, so the agonist response returns quickly. Other compounds dissociate more slowly, so it takes longer for the response to come back. The data are fit to a simple exponential equation to determine the antagonist residence times shown here. Now we're going to put all this together with a case study from drug discovery. This is from a project I was involved with. At Neurocrine Biosciences, in partnership with GSK, we were developing antagonists for the corticotropin releasing factor receptor. CRF is a stress hormone and neuromodulator implicated in neuropsychiatric disorders and endocrine diseases. Around 2003, we had advanced our first compound into the clinic, which showed efficacy in biomarker and early phase 2 studies. It was this guy on the left here, NBI 30775. Unfortunately, this compound was discontinued for tox reasons, so we were engaged in a backup campaign. What I'll show you here today is how residence time led to the development of our backup compound, Verusafon. The story of this work was recently published in this paper. Here's some data from the in vivo model we were using. What we're measuring here is inhibition of a biomarker in rats, adrenocorticotropic hormone, or ACTH. CRF stimulates ACTH release, which is inhibited by antagonists. And here's the data for our discontinued clinical compound, NBI 30775, showing nice strong efficacy in vivo. Here we're plotting inhibition of ACTH versus the plasma concentration of the compound. This is the in vivo assay we were using to identify advanced leads in our backup campaign. The in vitro affinity assay we were using to guide SAR was a radioligand binding assay run for 90 minutes at room temperature, so a pretty standard assay. Listed here is the affinity of 5 of the first compounds we pursued in the backup campaign. You can see their apparent binding affinity is similar to that of 775. Now, when we tested these compounds in vivo, we were surprised to see they were much less active than 775. You can see on the graph here they were much less effective, even though the plasma concentration of the new compounds was in many cases quite a bit higher. This was a problem. Our in vitro assay was not predicting in vivo activity of the compounds. We tested all the usual suspects to figure out what was going on here, but nothing could explain the results. It wasn't protein binding, for example. These compounds were much less protein bound than 775. This disconnect between in vitro and in vivo activity caused a lot of problems for the project, as you can imagine. Around this time, we were playing around with radiolabeled analogues, and we tested the binding kinetics of tritiated 775. We ran a dissociation assay, and were pretty surprised to discover the residence time was really long. At room temperature, the temperature we were using for affinity assays, there was little dissociation even after 5 hours. We extrapolated this out to be a residence time of about 3 days. As you can imagine from the previous slides, this has the potential to really mess with the affinity assay we were using, which was run for 90 minutes. This is shown in this simulation of the binding assay. The true affinity of 775 in black here is around 0.3 nanomolar. The affinity we measure at 90 minutes is way right shifted, coming in around 9 nanomolar. Under this condition, we were underestimating affinity by about 30-fold. Now it's worth pausing for a moment here and doing a quick thought experiment. Imagine if we were using this measured value of 9 nanomolar here in human dosing prediction. Imagine now the true affinity is 30-fold higher. This has the potential to dramatically impact drug effect in man. If there is any risk of target-based toxicity, failure to recognize the true affinity of the drug could result in overdosing and adverse events in safety trials. This really long residence time made it very difficult to measure affinity using a standard assay. We would have to incubate for 6 days to get a reliable number, and this was obviously not practical. So instead, we used an alternative approach. We actually measured the kinetics of the compounds, the K-on and K-off. Then we used that equation I showed you earlier to calculate the KD by dividing K-off by K-on. This gave us the true affinity of the compounds. This was done using a competition kinetics binding assay like we saw earlier. One final detail is we raised the temperature to 37 degrees. This decreased the residence time of 775 down to 4 hours, which made it measurable. Now, when we did this for our new compounds, we finally had an explanation for why they were so much less active. The true affinity of the compounds was much less than that of 775, as shown by the Ki values listed here. Under the conditions of the assay, 775 came in at 0.8 nanomolar. However, the new compounds were less potent. For example, compound 5 was 180-fold less active in terms of its true affinity for the receptor. This explains why the compound was less active in vivo. This provides a real-world example of how a long residence time can impact drug discovery. 775 was much more potent than we thought it was compared with the other compounds, owing to its long residence time. Now we had solved this mystery, we decided to use the kinetic Ki to triage for in vivo testing in order to find new active molecules. This is our in vivo rat ACTH model with 775 shown as before. We were able to find numerous compounds that were active in the model, as you can see. This compound in red here became our lead compound and was renamed Verusafont. We were happy to see this compound display clinical efficacy in a disease population. Here we are measuring ACTH in women with an endocrine disorder called congenital adrenal hyperplasia. These individuals produce too much ACTH, and Verusafont was able to reduce this, as shown here. This completes the story, showing how binding kinetics can have a real-world impact on drug discovery. Understanding the residence time solved a problem, the disconnect between in vitro and in vivo assay results, and this was then applied to the discovery of new molecules, which ultimately led to clinical proof of mechanism for the target. Now we will conclude with the question of when to measure binding kinetics in the drug discovery cascade. As you have seen, kinetics has major implications for the quantification of drug effect, but it does take a commitment of resources to evaluate the kinetics. A reasonable trade-off is to assess the kinetics of lead compounds, for example, at the advanced lead stage when we are starting to test compounds in vivo. This can avoid surprises of in vivo activity that can result from slow dissociation. This can be extended to selected compounds from hit to lead to see if there is a long residence time issue for the series. At this stage, qualitative assays can be used to test for a long residence time. This can be modified by circumstances. It may be decided before a project commences that a long residence time is a necessary feature for the therapeutic mode of action, and kinetic assays will therefore be built into the project plan from the start. At the later stages, binding kinetics is of high potential significance, and it is recommended that the residence time of the compounds be measured. The worst case scenarios of residence time are pretty bad here. For example, if the affinity has been underestimated owing to a long residence time, the target could be much more extensively occupied by the drug candidate than expected, with the potential for mechanism-based toxicity. If a short duration drug is the goal, a long residence time could dramatically distort the time course of efficacy, as we saw earlier. These issues could be prevented by simply measuring the residence time of the candidates earlier in the cascade. This brings us to the end of the presentation. I'd like to acknowledge some folks. On the left here is the CRF project team from Neurocrine and GSK, and on the right are colleagues and collaborators who have provided really useful feedback and discussion over the years on the kinetics topic. And finally, thank you very much for your time and attention.